Thank you for those great presentations. It's our pleasure to welcome Dr. Sruthi King to the panel for our Q&A session. Dr. King joined FDA in 2008 as a pharmacologist in the Division of Gastroenterology and Inborn Error Products within the Office of New Drugs, and later moved to the Office of Generic Drugs as a team leader in 2015, and now serves as an Associate Director of Pharmacology and Toxicology in the Division of Clinical Review within OGD. As a reminder, real-time questions to presenters can only be accommodated by those registered and logged in to the Adobe Connect application. Please register to gain access. We'll answer questions relevant to the content presented in this session as time allows. And also, to register for the April 9th Drug Master File and Drug Substance Q&A webinar, please go to fda.gov/CDER SBIA. Now moving into our Q&A panel session, our first group of questions will be directed to Dr. Gupta. And this is the first question. If the MDD is not available, how can we request this information from OGD?
Thank you for that question. If the MDD is not clear from the RLD label, you can seek input from OGD through a controlled correspondence, and I refer you to the controlled correspondence guidance for industry for further details. Thank you.
Thank you for responding to that question. The next question is also for Dr. Gupta. Where can we get reliable information on duration of treatment? Are there any regulatory acceptable sites available?
The duration of treatment for a drug can be determined through the RLD label or by doing a literature search to support the duration of use. However, this will be a review issue, and if you still have further questions regarding the duration of treatment, you can seek OGD's advice through a controlled correspondence, and I again refer you to the controlled correspondence guidance for industry for further details. Thank you.
Thank you for responding to that question. The next question. A generic drug substance itself contains an aromatic amine alerting structure and gives Ames positive alert in QSAR. However, it is known that is not genotoxic mutagenic and has lifetime duration of use. The RLD has the same route of synthesis. Its process-related impurity has the same structure, only instead of an aromatic amine, it contains an aromatic nitro, and also has a structure alert and is Ames positive in QSAR. How should we evaluate this nitro impurity? Is it possible to control this nitro impurity at 0.1% limit, or should TTC limit be applied?
Thank you for that question. In this case, since the alerting structure is different than the API, a QSAR should be conducted using complementary models as described in ICH M7, as well as by expert knowledge. And I refer you to Dr. Krulik's presentation for further details. If it is positive for mutagenicity, then it can be either controlled at the TTC or an Ames assay can be conducted. If it comes as negative in the Ames assay, this overrules the structural alert, and this impurity can be controlled as a non-mutagenic impurity per ICH Q3A or Q3B. Thank you.
Thank you for responding to that question. We have two more questions for Dr. Gupta. Are mutagenic evaluation and toxicological studies the responsibility of API DMF holders or of the ANDA holder?
The responsibility for the mutagenic evaluation and the toxicological studies is the responsibility of the DMF holder for the drug substance. Thank you.
Thank you for responding to that question. The last question in this round for Dr. Gupta is the following. When one should do a 14-day repeat dose toxicity study for impurity qualifications, what is the upper range of clinical duration to go with a 14-day toxicity study?
That's a good question. For drug substances which have duration of use less than or equal to 1 month, a 14-day toxicity study is sufficient. And I further refer you to Table 4 of ICH M7 to provide further guidance regarding the drugs which are in the category of less than or equal to 1 month duration. Thank you.
Thank you for responding to that question. The next group of questions will be directed to Dr. Johnson. And here's the first question. Could you please advise on how the total nitrosamine impurity limit should be defined?
That's an excellent question. So when you're looking at your drug substance specification, if you have more than one nitrosamine which is controlled, each nitrosamine may be controlled up to its acceptable intake. However, the total nitrosamine limit is 26.5 nanograms per day. That is just to ensure that we don't have multiple nitrosamines adding up to more than that amount. So individuals, each up to their acceptable intake, but if there is more than one nitrosamine controlled in the specification, the total nitrosamine limit is 26.5 nanograms per day based on the MDD.
Thank you for responding to that question. The next question, is there a mechanism for industry to alert the agency of any potential nitrosamine type of impurities and vice versa?
That's a good question. We have the DMF OGD mailbox. That's one route. There are also potentially other routes through the Office of Compliance or the Office of Surveillance. If you have a DMF that is currently under review with the agency, you may also send the questions again through that DMF OGD mailbox to us, and we can get back to you. If we find that there's other opportunities, we'll try and answer that in the April 9th follow-on.
Thank you for responding to that question. The next question. Is there any recommendation or requirement for the purity level of recovered solvents?
That's a really good question. So a recovered solvent should have purity that is, acceptable for use in your process. So we know when you have a recovered solvent, there's obviously going to be things in it that were not there in the fresh solvent. Most of those will be other processed solvents. There could be intermediates or starting materials that carry over. So the better question is to understand what is in that recovered solvent. Please remember that ICH Q3C does not have any impurity at not more than 0.10%. That's for ICH Q3A. So when you're looking at recovered solvent, you really just need to be looking for peaks that are showing up continuously, especially if they're about the same size as some of the other recovered solvents. If they're peaks that are related to impurities that are controlled under other tests, then you're fine. If they're processed solvents, that's fine. And as I said before, if they're byproducts of two solvents reacting, you're fine. It's those little peaks that are consistently showing up that you're not sure what they are. So the answer is your recovered solvent needs to have a purity that's acceptable for use, and it's going to require some investigation on your part about what that means for you.
Thank you for responding to that question. The next question. For method validation by LCMS or an equivalent instrument, what would you consider an acceptable recovery for MDMA or other nitrosamines in an accuracy recovery study?
Okay, another excellent question. Generally, because we know that these nitrosamines have fairly low limits, we generally expect to see your accuracy to be somewhere between 70 and 130%. If it gets outside that range, then we're a little concerned because most of these are being measured in samples that are fully dissolved, and there should not be any kind of extraction issues. So generally, we typically see and kind of expect that your accuracy values should be between 70 to 130%. If they're extremely outside that range, then we would probably expect an explanation for something that fell outside that.
Thank you for responding to that question. The next question, and we have a couple more. What is the acceptable limit of sodium nitrite in sodium azide? How do we determine it?
Another excellent question. So we know that sodium azide is kind of a hidden source of sodium nitrite, and the limit that you may want to put on that is going to be very dependent on your process. If that nitrite is the potential source for a nitrosating agent in your process, you're going to have to determine how much your process can handle in order not to generate nitrosamines that either are above a level that carry into your process or wouldn't exceed the AI if there is a limit for them in the specification. So that is going to be very dependent on your process. We've seen sodium azide that contain from some suppliers that contains as low as 50 to 60 ppm, all the way to some suppliers that have well over 1,000 ppm. You will have to determine how much your process can handle and then work with a supplier that can provide a reagent of appropriate quality.
Thank you for responding to that question. We have just a couple more for Dr. Johnson. The FDA September 2020 guidance, Control of Nitrosamine Impurities in Human Drugs, mentions that API manufacturers should use sensitive methods with an LOQ that is less than 0.03 parts per million to analyze the content of nitrosamine impurities in their API. In cases where this is not possible, is it acceptable to use methods with an LOQ that is 30% of the limit of the nitrosamine impurity as per the EMA guidelines for nitrosamine impurities in human medicinal products?
Yeah, thank you for that question. We do recommend that LOQs typically be 0.03 ppm because most products are requiring a limit that is sub 1 ppm. And in order to accurately quantitate that, you do need a lower limit of quantitation. This is especially important if you have to do the total nitrosamine. In order to quantitate each one of those individual nitrosamines, you will need a sufficiently low limit of quantitation. However, if the limit of your nitrosamines based on the MDD of the drug product is higher than 1, let's say it might be 5 or 6 ppm, then you might be able to argue that a higher LOQ than 0.03 would be appropriate. If you cannot achieve that, then please explain your rationale and then we can evaluate it. But in general, we do encourage you to get down to 0.03 ppm, and most people have seemed to be able to do that. Again, if you run into questions, please contact us and we'll try and work with you.
Thank you for responding to that question. The final question for Dr. Johnson in this round. Is the nitrosamine risk assessment needed in the DMF?
That is a very good question. And the answer is no. The nitrosamine risk assessment should be kept on site. However, if after you've done your risk assessment, there have to be changes to your process or any control strategies, those should be reported to your DMF. If you discover that you do have nitrosamine impurities, any limits that you propose or control strategies should be justified with data, and that should be reported to your DMF. But the entire risk assessment does not need to be supplied to your DMF.
Thank you for responding to that question. The next group of questions will be directed to Dr. King. And this is the first question. The FDA September 2020 guidance, Control of Nitrosamine Impurities in Human Drugs, specifies the ADI for 6 nitrosamine impurities. For nitrosamine impurities whose acceptable daily intake, or ADI, is not known, what considerations are to be taken into account while setting the limit? Can we assume a class-specific ADI of 26 nanograms per day based on the ADI for the most potent nitrosamine as per part of the guidance? Or should we consider 18 nanograms per day as per the EMEA nitrosamine guideline on impurities in human medicinal products?
Thank you for that question. It's a very good question, and yes, we do look at available information for the nitrosamine when establishing acceptable intake limits. The approaches for calculating an acceptable intake limit are described in ICHM 7. When data are not available to calculate an acceptable intake, and by this I mean mutagenicity data or rodent carcinogenicity data for that specific nitrosamine, sponsors can consider approaches whereby they identify a surrogate compound. And if such an approach is being taken, we advise that DMF holders or ANDA applicants submit those justifications and rationale to the agency for review. Now, we do interact with our international regulatory colleagues, and at this time FDA is not applying a class-specific limit for nitrosamines. We do refer you to the guidance, the FDA guidance that describes the allowable total nitrosamine intake when setting limits for your drug product or drug substance. Thank you.
Thank you for responding to that question. The next question. How did FDA select the nitrosamines listed in the guidance? Are these the only nitrosamines we should consider?
Thank you for that question. So FDA conducted risk assessments as they became aware of presence of specific nitrosamines in drug products. And so the acceptable intakes posted in the guidance are based on those risk assessments and root cause investigations. And as we conducted our root cause investigations and were reviewing information submitted from sponsors, we then also included additional nitrosamines into that acceptable intake calculation. And so the ones in the guidance are not the only ones that you should be looking out for. You should be looking at the FDA guidance and following the risk assessment procedures described in the FDA guidance. And for those nitrosamines that are not listed in the guidance, you can use approaches described in ICHM 7 to calculate appropriate acceptable intake limits.
Thank you for responding to that question. The next question. How does FDA identify interim limits? Why are interim limits not recommended for all drugs?
Thank you for that question. FDA has identified interim limits for specific nitrosamines, and this is recommended based on the drug that is being impacted by the presence of that nitrosamine. Medical necessity is a big factor when considering interim limits for a specific nitrosamine. In general, the acceptable intakes for each nitrosamine consider a lifetime of exposure. And while M7 describes risk assessments which consider the actual duration of exposure and allows for adjustments for shorter exposures, interim limits are are described where flexibility is required to maintain access to a specific drug product, a specific medically necessary drug product. And it is considered a short-term strategy while sponsors are able to achieve the lifetime acceptable intake for that nitrosamine.
Thank you for responding to that question. Could you tell us, how does FDA calculate acceptable limits for nitrosamines?
Thank you for that question. FDA uses approaches described in ICHM-7 when calculating an acceptable intake limit for nitrosamines. As I described, nitrosamines belong to a cohort of concern group of compounds because of their high potency for carcinogenicity. Many are mutagenic and carcinogenic. They are potent rodent carcinogens and probable human carcinogens, and therefore additional care and tighter control limits are applied to nitrosamines. When calculating an acceptable intake limit, we look at available rodent carcinogenicity data and consider the TD50 approach when calculating the acceptable intake. And the formula for calculating acceptable intake is provided in ICHM-7. When considering whether rodent carcinogenicity data for a specific nitrosamine are appropriate, we look to see how robust the data are. And in determining whether the data are robust, we consider the number of animals in a dosing group, the treatment regimen, the types of data that are reported in the study, how many studies are available, etc. So depending on our determination of whether the data are robust, we then look to identify a TD50 and calculate the acceptable intake as described in ICHM-7.
Thank you for responding to that question. And the last question for Dr. King in this session: does FDA consider how the product is used, for example, short versus long-term exposure, when setting limits for nitrosamines? Is FDA considering less than lifetime exposure when it comes to nitrosamines, and if not, why?
That's a very good question. FDA does acknowledge that the less than lifetime approach is described in ICHM-7 and does allow for adjusting exposures to compounds based on how they are used. However, nitrosamines belong to a group of highly potent mutagenic carcinogens. As I described, they're part of the cohort of concern, and multiple nitrosamines have been shown to induce tumors in multiple species at relatively low doses and often after very short durations of dosing, sometimes even single doses. Therefore, a simple adjustment to identify a less than lifetime limit would be difficult to make given the uncertainty in how the cancer risk of cohort of concern carcinogens would change with the shorter duration of exposure. And therefore, in the absence of that specific information to address these unknown risks, less than lifetime adjustments are not recommended for nitrosamine impurities. Thank you for your question.
Thanks for responding to that question. The next group of questions is for Dr. Gupta, and here's the first question. I have a question about an impurity which is a reproductive toxicant. TTC is used as the control limit. However, the dietary cumulative estimated daily intake is thousands times lower than TTC. Is CDI applicable for drug products, or is TTC more appropriate?
That's a great question. For impurity qualification, both the mutagenicity and general toxicity of the impurity should be evaluated. We typically do not rely on dietary exposure levels for safety evaluation of drugs. If an impurity is non-mutagenic, it can be controlled as per ICHQ3A, And in this particular case, if the impurity is a reproductive toxicant, then a compound-specific limit should be calculated based on the available reproductive and developmental toxicity studies. And this will be a review issue when the justification comes in with the ANDA. Thank you.
Thank you for responding to that question. The next question for Dr. Gupta. In the example of different routes of administration, is the one-off high priming intravenous dose assumed to be the daily dose for chronic use, even if it is only administered once at the start of treatment? And does the route of administration have to be considered when determining the worst case scenario?
For the first part of the question, yes, the intravenous dose should be, the high intravenous dose should be considered for setting limits for non-metagenic impurities, because such impurities can manifest their effects even after acute exposure. And yes, the route of administration and the context of use of the drug should be considered in addressing the safety of the impurity. Thank you.
Thank you for responding to that question. Next question is for Dr. Gupta. If in an Ames assay at the top dose level no positive result is obtained, do we need then to test at a higher dose for confirmation?
The top dose recommended by the OECD 471 guideline is 5,000 microgram per plate if the test article does not precipitate or is not cytotoxic. If the Ames assay is fit for purpose and is conducted appropriately and it does not show increase in revertant colonies as compared to the control, then there is no further need to test at higher doses. Thank you.
Thank you for responding to that question. The next group of questions will be directed to Dr. Johnson. I carried out nitrosamine research when I was a postdoc at NIH NCI Bethesda in 1981-83. My mentor has an instrument, TEA, specific for nitrosamine. Does FDA recommend TEA?
That's a good question. That's actually a method that we really haven't seen here at FDA. However, we don't specifically tell firms what analytical methods they can use to control their nitrosamines. However, if you use that kind of method, we would expect a full method validation report to be sent. But we may have to consult that out to our colleagues in Office of Testing and Research because they are the experts on that kind of method. But yes, you may choose the method that you want to control them, but for that kind of method, we probably would need to get some input from our colleagues.
Thank you for responding to that question. The next question, what is the acceptable limit of sodium nitrite in sodium azide?
Yeah, I think I had touched on that on the last one. But again, it really depends on your process. If the nitrite has the potential to create a nitrosamine in your system, then the amount of that nitrite coming in, you will have to determine that probably through a spike purge. It would be my best advice.
Thank you for responding to that question. The next question, can purified water be the source of nitrosamine impurities?
That's a great question. I'm actually seeing several questions about water, purified water, potable water. You know, that is a hard question to answer. Every country has their own requirements for water quality. We do expect, water to be of, equal quality to like for purified and potable water that is, in the USP. But whether or not that is a risk really is going to depend on your location. If you think that that's, something that might be showing up, then it's probably worthwhile in, looking at that to see if it's an actual risk. But that's actually kind of a hard question because it's so kind of location dependent.
Thank you for responding to that question. The next question, does the FDA guidance apply to biologics?
Okay, great question. And the answer to that is no.
Thank you for responding to that question. Looks like we have time for just one more question for Dr. Johnson. In the case of multiple nitrosamines, what is recommended as the limit for each individual nitrosamine, understanding that the total should be less than 26.5 nanograms per day?
So, in the case where you're controlling multiple nitrosamines in your specification, you are allowed to have the limit based on the AI and the MDD for each nitrosamine. And you have that separate requirement for total nitrosamine. So you may have the AI for each one, but the total nitrosamine acceptable intake is 26.5 nanograms.
Thank you for responding to that question, and thank you to our panelists for answering all of the questions that came in. We'll now